In the hushed, sterile expanse of a high-security paleogenetics laboratory in Stockholm, a discovery was made that defied the fundamental laws of molecular biology. For decades, the scientific community has held a central dogma as immutable as gravity itself. DNA endures, but RNA vanishes. We have always viewed DNA as the stone tablet of the genome, capable of surviving for over 1 million years in the right conditions. RNA, on the other hand, is the fleeting messenger, the whisper. It is a fragile, transient molecule that typically degrades within minutes of an organism's death. To search for ancient RNA was considered a fool's errand, a pursuit of ghosts. But on a cold day in late 2025, a team of researchers led by Dr. Love Dal N and Dr. Emilio Mrmolsnchez shattered that dogma. They didn't just find a few fragmented scraps of genetic code. They recovered a coherent biological narrative from the muscle tissue of a woolly mammoth that died 39,000 years ago. This was not merely a sequence of nucleotides. It was a snapshot of a living, breathing creature's final moments. The RNA sequences they decoded told a story of terror, exhaustion, and biological stress. They revealed the metabolic engines firing in the legs of a juvenile mammoth fleeing for its life across the Pleistocene tundra. They corrected the animal's sex, rewriting the history of a specimen scientists thought they knew. And, perhaps most profoundly, they unlocked a new dimension in the quest to de-extinct the woolly mammoth, offering the missing instruction manual for how to turn a genome into a living giant. This is the story of Paleo-Transcriptomics, a revolutionary new field that has turned the frozen graveyards of the Arctic into a high-fidelity archive of Earth's lost history. To understand the magnitude of this breakthrough, we must first appreciate the chemical fragility of the molecule in question. Ribonucleic acid, or RNA, is the working class of the cell. If DNA is the master architect's blueprint stored safely in the vault of the nucleus, RNA represents the fleet of contractors, messengers, and builders that carry those instructions out to the construction site of the ribosome, where proteins are made. By design, RNA is unstable. It is meant to be produced, used, and recycled rapidly to allow the cell to react to its environment. If there is a sudden spike in temperature, the cell pumps out heat shock RNA. If there is a drop in blood sugar, metabolic RNA profiles shift in seconds. Because of this, RNA is chemically prone to hydrolysis, a reaction where water molecules tear the chain apart. Furthermore, the world is awash in enzymes called RNases, which evolved specifically to destroy free-floating RNA to prevent viral infections. In a decaying carcass, these enzymes usually scrub the cellular hard drive clean within hours. As Dr. Dal N explained, the team suspected that the main reason RNA degrades is due to these RNase enzymes. They figured that if they could sample mammoths that had frozen quickly after death, there might be a chance. The specimen at the heart of this revolution is a juvenile woolly mammoth named Yuka. Discovered in 2010 on the mist-shrouded coast of Oyogos Yar in northern Siberia, Yuka is widely considered the best preserved mammoth ever found. The animal was roughly 6 to 8 years old when it died, encased in permafrost that acted as a natural time capsule. Yuka was not just a pile of bones. The carcass retained strawberry blonde fur, intact skin, and, crucially, soft tissue that had been desiccated and frozen rapidly. The preservation was so exquisite that when the carcass was first examined, the brain was found intact inside the skull. But while Yuka's DNA had been sequenced years prior, providing the static code of its identity, its RNA remained an untapped reservoir of functional data. The decision to hunt for RNA in Yuka was a massive gamble. Dr. Mrmolsnchez and his colleagues at the Center for Paleogenetics and the SciLife Lab in Sweden knew the odds. Previous attempts to recover ancient RNA had been limited to much younger samples, such as a 130-year-old thylacine skin and a 14,000-year-old Pleistocene wolf. A 40,000-year-old sample was a leap into the deep time of the unknown. The success of the extraction hinged on the unique physics of the Siberian permafrost. The conditions that held Yuka were not just cold, they were stable. The carcass had likely been buried and frozen shortly after death, locking the water molecules in its cells into a rigid lattice before they could participate in the hydrolysis reactions that destroy RNA. This state, known as the glass transition, effectively halts biological time. Inside Yuka's muscle cells, the RNase enzymes were frozen mid-attack, and the fragile strands of messenger RNA were trapped in a molecular suspension. For nearly 400 centuries, while civilizations rose and fell and the ice sheets retreated, those strands waited. However, recovering the RNA was only half the battle. The true challenge lay in proving that the sequences were authentic and not modern contamination. In the world of ancient DNA, contamination is the arch-nemesis. A single flake of skin from a lab researcher or a droplet of aerosolized bacteria can drown out the faint signal of the ancient molecules. The team employed a suite of high-tech forensic methods to authenticate their catch. This was not standard sequencing. This was molecular archaeology performed with the precision of a diamond cutter. So, how do you tell the difference between a strand of RNA from a 40,000-year-old mammoth and a strand of RNA from a bacterium living on the sample today? The answer lies in the damage. Over thousands of years, ancient genetic material undergoes specific patterns of degradation. The most telling of these is a process called cytosine deamination. Over time, the chemical base cytosine loses an amine group and turns into uracil. Since uracil is found in RNA naturally, this might seem confusing. However, in DNA, this change manifests as a mutation during sequencing. In ancient RNA, the specific accumulation of these errors at the ends of the molecules acts as a molecular timestamp. Dr. Marmolsn's team looked for these specific lesions at the ends of the RNA fragments, and they found them. The RNA sequences from Yuka showed the classic statistical curve of cytosine deamination that characterizes true ancient material. It was the molecular equivalent of carbon dating, an undeniable signature of antiquity. Because Yuka's RNA was fragmented into tiny pieces, often shorter than 50 nucleotides, the team used a specialized protocol optimized for short, single-stranded molecules. By treating the samples with enzymes that digested double-stranded DNA and focusing on the single-stranded RNA, they enriched the sample. They then used bioinformatic filters to digitally scrub the data, comparing every sequence against the genomes of modern elephants, humans, and bacteria. Only sequences that aligned perfectly with the elephant lineage and showed the characteristic ancient damage patterns were kept. The result was the world's first paleo-transcriptome. Now, what does 40,000-year-old RNA tell you that DNA cannot? DNA is simply a list of ingredients. It tells you that the mammoth had the genes to make muscle, hair, and hemoglobin. But it doesn't tell you how much of each ingredient was being used, or when. RNA is the chef cooking the meal. It reveals which genes were switched on at the exact moment of death. When the team analyzed the gene expression profiles from Yuka's leg muscle, they didn't just see a static list of genes. They saw a physiological drama unfolding in real time. One of the most striking findings was the upregulation of genes associated with cellular stress and metabolic exhaustion. Specifically, the researchers identified transcripts for the gene ANKRD1, which is known to play a critical role in muscle response to stress and injury. In modern animals, ANKRD1 levels spike during intense physical exertion or trauma. The presence of these transcripts in high abundance suggests that Yuka was not resting peacefully when he died. His muscles were working overtime, flooded with the molecular signals of exhaustion and repair. This molecular data aligns chillingly well with the physical forensic evidence on the carcass. Yuka's body bears deep, unhealed scratch marks on the hind legs, scars that match the claw spacing of the cave lion, the apex predator of the Pleistocene steppe. Putting the RNA and physical evidence together, a harrowing narrative emerges. A young mammoth, separated or straggling, is ambushed by lions. He runs, his muscles screaming with exertion, the ANKRD1 gene firing frantically to manage the stress. He survives the initial attack, but, exhausted and wounded, perhaps retreats to a shallow pond or falls into a mud trap where he eventually succumbs. The freezing mud locks his muscles in that state of metabolic panic, preserving the chemical scream of his final flight for 40,000 years. Beyond the drama of his death, the RNA provided a blueprint of how a mammoth lived. The transcriptome revealed a dominance of isoforms associated with slow-twitch muscle fibers. These are the endurance engines of the animal kingdom, efficient and fatigue-resistant. This makes perfect evolutionary sense for a woolly mammoth. These animals were not sprinters. They were long-distance wanderers, built to march endlessly across the vast arid mammoth steppe in search of sparse vegetation. The RNA data confirmed that Yuka's physiology was tuned for efficiency and heat retention, rather than explosive speed. Additionally, the RNA sequencing settled a long-standing debate regarding Yuka's sex. For years, scientists referred to Yuka as a female, based on a visual inspection of the preserved genitalia. However, mummified soft tissue can be deceptive due to distortion. The team found transcripts from the DDX3Y gene and other Y chromosome-specific loci, which are only present in males. Yuka was, indisputably, a male. This finding highlights the power of Paleo-Transcriptomics to correct the archaeological record where physical preservation fails. Perhaps the most scientifically significant discovery in the Yuka study, however, was not the protein-coding RNA, but the microRNAs. If mRNA is the contractor building the house, microRNA is the building inspector who can shut down construction or modify the blueprints on the fly. These tiny molecules regulate other genes, dimming their expression or silencing them entirely. They are the master controllers of the genome. The team recovered hundreds of microRNAs from Yuka. Most were identical to those found in modern elephants, highlighting evolutionary stability. However, they also found novel microRNAs, genetic regulators that seemingly do not exist in modern elephants or humans. This is a revelation. It suggests that what made a mammoth was not just having hair genes or fat genes, but having a unique set of regulatory instructions that told those genes how to behave in the cold. For example, a specific microRNA might have dampened the expression of heat-sensing genes, allowing the mammoth to tolerate freezing air without shivering. Another might have upregulated insulin signaling to promote the rapid deposition of brown fat. Finding these lost regulators is like finding the source code for an extinct operating system. The sequencing of Yuka's RNA marks the maturation of a new era in paleontology called multi-omics. Traditionally, we studied fossils with our eyes. Then came the era of ancient DNA. Now, by combining genomics, transcriptomics, and proteomics, we can build a holographic view of an extinct life. We are moving from describing bones to simulating biology. This also opens the hidden world of paleovirology. One of the most tantalizing prospects is the potential to find ancient RNA viruses. Many of the world's most dangerous pathogens, like influenza and measles, are RNA viruses. Because RNA degrades so fast, the history of these diseases has been a black hole. While the Yuka study did not report a specific active viral infection, the successful recovery of endogenous mammoth RNA proves that viral RNA could survive. We can now hunt for the ghosts of the Ice Age's plagues to test theories like the hyperdisease hypothesis, which suggests that novel pathogens wiped out megafauna populations. The implications of this discovery for colossal biosciences and the broader de-extinction movement are staggering. Until now, the plan to bring back the woolly mammoth has relied on cut-and-paste genomics, taking the genome of an Asian elephant and editing in mammoth traits. But having the genes is not enough. You need to know how to regulate them. Imagine trying to build a medieval cathedral using a blueprint for a modern office building, with only a bag of loose gargoyles to add on. You might stick the gargoyles on the walls, but without the architectural logic, the structure might collapse. The RNA data from Yuka provides that architectural logic. It tells scientists how much expression is needed for cold-adapted genes. It allows for fine-tuning CRISPR editing to swap promoter regions, ensuring high expression of stress response genes like ANKRD1. It highlights the need to engineer specific microRNAs into the hybrid embryo to ensure it develops correctly. The Yuka RNA data moves the project from a genetic approximation to a functional resurrection, allowing the creation of an animal that doesn't just look like a mammoth, but metabolizes like one. The success of the Yuka project has kicked the door open for a new wave of exploration. If RNA can survive for 40,000 years in permafrost, where else might it be hiding? Dr. Dal N and his team believe that in the deep, stable permafrost of northern Greenland or Antarctica, RNA might survive for hundreds of thousands of years. We could potentially recover the transcriptomes of pre-human hominids or ancient polar bears. Furthermore, this impacts museum collections globally. The study suggests that desiccation can also preserve RNA, meaning the thousands of dried skins and mummies in museums could be goldmines of transcriptional data. We could sequence the RNA of the dodo or the passenger pigeon, understanding their biology and stress levels at the time of their extinction. Of course, this brings ethical questions. We are now peering into the intimate biological moments of creatures that died millennia ago, reconstructing the suffering of a calf chased by lions. There is a responsibility in how this data is used to ensure we create functional, healthy animals capable of ecosystem restoration, rather than suffering curiosities. The sequencing of 39,000-year-old mammoth RNA is more than a technical triumph. It is a conceptual shift. For centuries, the word extinct meant gone, erased, silent. Paleo-Transcriptomics tells us that the voices of the past are not silent. They are just waiting for us to learn how to listen. Yuka the mammoth has spoken to us across the chasm of 40 millennia. He has told us of his sex, his muscles, his terror, and his cold-adapted resilience. As we stand on the brink of potentially returning his kind to the tundra, we do so armed with a new depth of understanding. The ice is melting, revealing its secrets, just as we develop the tools to read them. In the strands of fragile, ancient RNA, we have found a way to cheat death, turning the frozen graveyards of the past into the laboratories of the future. The ghosts of the Ice Age are no longer just bones in the ground. They are data, they are stories, and soon, they may be alive again.